Hello everyone, my name is Shraddha and welcome to my YouTube channel Node of Biology. The topics for today's discussions are vesicles, peroxisomes and endosomes. For future videos, please like, share and subscribe to my channel Node of Biology. Vesicles. Vesicles are small membrane enclosed transport units which can transfer molecules such as proteins, hormones, etc. between different compartments within the cell or outside of the cell. Majority of the transfer that occurs in a cell occurs between the endoplasmic reticulum to Golgi apparatus and then to different locations. Every vesicle type has a function to perform, and each vesicle is necessary for different kinds of biological process. Vesicles can be divided into two types: transport vesicle and secretory vesicles. Transport vesicle. The function of the transport vesicle is to carry molecules around the cell. The structure of the transport vesicle is similar to that of any biological membrane found in a living organism. That is, it is made up of phospholipid bilayer, as we can see in the illustration on the right side of the slide. The transport vesicles primarily performs two things. Either it takes the protein synthesized by the ribosomes to the ER and then to the Golgi apparatus for further sorting, or the Golgi apparatus can, once after receiving the protein, can pack it into the secretory vesicle, and then these secretory vesicle can release their content, or that is the protein, outside of the cell. The other type of vesicle is the secretory vesicle. It is a membrane enclosed organelle which brings materials, that is the protein, hormones or antibodies, to the plasma membrane so that they can then be released outside of the cell through a process known as exocytosis. As we can see in the illustration above on the slide, secretory vesicle containing whatever could be a protein or hormone or antibodies is brought closer to the plasma membrane, and this vesicle then fuses with the plasma membrane, thereby releasing its content outside of the cell. Peroxisomes are found in the eukaryotic cells. They are small membrane-bound structures which contain a variety of enzymes, such as digestive enzyme that can break down toxic materials within the cell, and oxidative enzymes which are used in metabolic activity. The peroxisomes can break down reactive oxygen species such as hydrogen peroxide into water and oxygen. They are self-replicating organelles, and in plants, the peroxisomes in the form of glyoxysomes convert the stored oil into molecules that the plant can use for photosynthesis. Endosomes. Endosomes are membrane-bound cytosolic vesicles found in animal cells, which are formed during the process of endocytosis. To learn more about endocytosis, please follow the link in the description box. The endosome directs the vesicle by either fusing with them or pinching the plasma membrane. The internalized material usually contains nutrients in the form of proteins or pathogens such as bacteria. Once the material is engulfed into the cell, the endosome then starts sorting of the material within the cell or within the subcellular compartment, and thus the endosome is responsible for controlling protein and lipid trafficking among the other subcellular compartments of the cell. For future videos, please like, share and subscribe to my channel Node of Biology.